Hey everyone! Today I'm going to teach you about endomembrane trafficking. Endomembrane trafficking plays a major role in maintaining homeostasis, growth and development, hormonal signaling, and adaptation of eukaryotes to how constant the environment changes. To start off, I'm going to begin with protein synthesis, which is the process through which a cell generates new proteins. This process includes going through transcription, translation, and post-translational modification. These proteins are made using information provided by DNA found in the nucleus. The nucleus is the central or core of a cell that contains the cell's hereditary information and controls a cell's reproduction and growth. The nucleus is enclosed by a nuclear envelope, and materials exit through the nuclear pores into the cytoplasm. DNA, deoxyribonucleic acid, is contained within the nucleus and stores genetic code within chromosomes. A molecule of DNA is made from two DNA strands and is paired by hydrogen bonding by nitrogenous bases. These nitrogenous bases include adenine, thymine, cytosine, and guanine. Adenine and thymine pair, while cytosine and guanine. mRNA carries info from DNA outside the nucleus to continue the process of protein synthesis. RNA replaces thymine base with uracil. mRNA is formed through the transcription of DNA. Transcription takes place in the nucleoplasm. This process allows instructions for protein formation to be copied from DNA to mRNA. First, the DNA molecule is broken apart by RNA polymerase. As the template strand is zipped away, RNA nucleotides find complementary bases on the coding DNA strand. Each DNA triplet corresponds to a sequence of 3 nucleotides of mRNA called a codon. After mRNA strand is made, it detaches and exits through a nuclear pore into the cytoplasm where translation. After the mRNA strand exits through a nuclear pore into the cytoplasm, translation will begin. Through translation, the mRNA strand will attach itself to a ribosome. Then, tRNA will come in and attach the anticodons to the codons on mRNA. Here you will see another tRNA coming in and attaching its anticodon to a codon onto the mRNA strand. Here, once another tRNA comes in, the amino acid from the original tRNA strand will detach and attach itself to the next amino acid on the tRNA strand. tRNA will continue its process across the mRNA strand until a stop codon discontinues the process and a polypeptide chain is released. After the polypeptide chain is released, it goes out of the ribosome and a new protein is released. Post-translational modification. After the new protein is released, the polypeptide moves into the rough ER where sugars are added and the protein folds. The protein is then transported by a vesicle into the cis-phase of the Golgi apparatus. After transporting the protein by a vesicle into the cis-phase of the Golgi apparatus, the Golgi then matures to become the medial Golgi. Next, the proteins are transferred from the trans-Golgi or transphase and are moved to the plasma membrane. After the protein is transferred from the trans-Golgi or transphase and moved to the plasma membrane, the membranes then fuse together and the protein is let out of the cell. This is also known as exocytosis. And this concludes my presentation of endomembrane trafficking. Thank you very much.